warm welcome to Jumai!
So much excitement. Yeah, welcome to the knitting session. I hope you have your balls of wool. Let's start knitting. No. My name is Jumai. I am in the School of Engineering. I'm supposed to be an engineer, but more of a biologist. So I'm kind of a confused person. Who am I? Everyone goes through that state in your life. They're like, who am I? Loads of people go to like their bathroom and cry because they're like, I have no clue who am I, what am I supposed to do with myself? As a biologist, I'm gonna answer by just saying, you have 37 trillion cells in your body. An average individual has 37 trillion cells. Basically, if you put the cells side by side and line them around the earth, they're gonna go around 19 times. And that's what you have inside of you. That's who you are. I'm so glad Elmi went right in front of me because she spoke about the central dogma and how DNA goes to RNA, goes to protein, and now the dogma is very dogmatic. Well, we're all made up of these chromosomes. Basically, there are 22 autosomes, and there is the X and Y chromosome. And that is what, causes a lot of confusion in the world. So we all have 22. But this 23rd one is why some people are guys and some people are girls. And I have 2 X's, so 2 big ones. And the men have 1 big one and 1 tiny one. Cool. So basically, this guy's with 1 big one and 1 tiny one. Watson and Crick, in 1953, discovered the structure of the DNA. And basically, it's written in 4 letters, G, A, T, and C. And that's all the complexity that we have. And Elmi spoke about how we have about 3.2 billion nucleotides in us. And some other people, and she spoke about worms and stuff like that. So the Human Genome Project was completed in 2003. 2001, they said they had a draft. In 2003, they had a complete draft. And then at some point in time, they just wanted to have so many parties, so they kept releasing different copies of the draft. But it was from 1988 to 2003, and they spent $3.8 billion. But basically, when you get the result back, it looks like that. Oh, it's a gibberish. You go like, I want to sequence my genome. It's so cool. I'm going to get my genome. And then you get that. And you're like, I have no clue what this means. Do not worry, some people are paid to do that. So basically you have this gibberish made up of G, A, T, C, G, T, C, A, G, 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 whatever. And a lot of it is junk, and some of it gets translated, and all the yada yada. But what happens with this GATC that seems not to make a lot of sense is that when there are mistakes in this GATC that makes up you, whether it's a deletion, whether it's an insertion, whether it's a substitution, or whether things get duplicated and you have 3 copies of something when you should have 2 in the genes, you can have sicknesses. And some of us are affected by these sicknesses. Somebody has a grandma that has cancer, or someone has a brother that has Down syndrome, or someone has polydactyly. That might be cool if I had 6 fingers, but I have 5. But there are so many problems that can come from these mistakes, and they affect us. They might not affect you individually, it affects some of us, but it affects our families, it affects our community and our environment, and it's important for us to know what to do about them. So your genome is complex, it's unique to you. You can pass it on to your children. If you have a problem that runs in your family. So my parents, they are currently pre-diabetic. And I know that later on in life, I have a very high chance of being diabetic. So I have to be very careful about what I eat, what I put in my body. It's good to have that information, but some people are orphans, they don't even know. So we're now in the age where you can sequence your genome. And I checked yesterday, apparently the cheapest right now is about 1,850 pounds for a full genome. Well, I'm not sure they'll give you a full genome. Yeah, so the genome tells us a lot of things, but there are bad guys out there. As if we don't have enough problems of our own, we still have viruses. These viruses come, they don't just visit, Someone spoke about malaria. A mosquito bites you, you get infected. I've had malaria loads of times. I'm not dead, obviously. But the mosquito bites you, you get infected, it goes away. But you get viruses that actually integrate into the genome. They become a part of your genome. And you all have you have tiny balls of wool on your table. And those are infected genomes. So basically, you're thinking about your genome and 3.2 billion nucleotides inside of your nucleus, inside of each cell, each of the 37 trillion cells inside of your body that can wrap around the world 19 times. So we have those viruses integrated into the genome. And you have scissors, so you can pick a scissors. Some random person on the table, the least drunk one, can have a scissors and try to cut out this part of the genome, try to cut out the infected part. So what HIV does is, I'm not going to go into the cycle because everyone by this time of the night is not ready to go into how does it go in, how does it multiply, how does it become plenty. In summary, HIV goes in. It takes over. It just takes over the system and causes your cell to make multiple copies of it, and then it keeps making itself. But we have all over the world, we have about 35 million people currently living with HIV. A lot of them are untreated. A lot of them are in sub-Saharan Africa, where I come from. In the UK, there are about 100,000 people living with HIV. 2/3 are men, and then 1/3 are women. I like that. But okay, I don't like the fact that people have HIV. But 2/3 are men, the ones with the one big one and one tiny one. But there is antiretroviral therapy. So people use drugs, and the drugs kind of tackle different parts of how HIV multiplies, how it goes into the genome. So it affects, the drugs tend to tackle the parts, the active parts. So if you have a child in your house that runs around all the time, you can say, go to your room, that kind of thing. So that's what the drug does. The drug kind of stops it from multiplying, from causing chaos. But it doesn't eliminate it. Much like this picture, you have a child just on a bed, but there's a monster under the bed. It's silent, but it's lurking. And when it's time, when the child falls sick, when someone who is using so many drugs, so much of the antiretroviral therapy, the drugs they take, when the person feels like, I am confident enough, I'm alive, I can survive, I can thrive, I can live with HIV, say they fall sick for a few minutes, it can multiply again, So that's the problem with latent HIV. The HIV is just in the genome, it's right there, it's lurking. It's not multiplying, but it's there. It's waiting for the right moment to start up again and to grow and to grow and to grow until the person has AIDS, until the person cannot survive as a viable member of the community anymore, until they die. We don't want that. So, this is where I come in. Much like Tinkerbell, I tinker with nature. I work in a field called synthetic biology, and it's kind of like the interface between engineering and biology. Sometimes there are engineers who try to do biology, there are biologists who try to do engineering. And what I do is I just apply random and rational engineering principles, try to tinker with nature, with these genes, and say, how do I change things? How do I remove things from the genome? And I work with some enzymes called recombinases. I just sprinkle fairy dust, and then they change. No. I go to the lab, and then I work with these enzymes. They're called recombinases. Naturally, they exist in some cells, and what they do is they recognize specific regions of genomes, and then they cut it out. So what I'm doing is trying to get them to recognize HIV genome, and then cut it out, and then tape it back together. So the big question for the current stage of my PhD is, can we cut out HIV? Can we chop it out? If we have an infected genome, can we cut it out? And I have this kind of model. Say you have this big part. Okay, cool, cut it already. Say you have this big genome that has the red bit, which is the infected part. I can simply remove it and then join it back together using my enzyme. And then I have a tiny bit, which is just like the tiny bit of HIV left, which is not active. And because the HIV is not part of the cell anymore, it can multiply, it can do anything. And I'm using E. coli currently to do my work, so I'm not doing this in human cells, rat cells, any kind of thing that is scary. Just to put it out there. I'm making progress, as are all the researchers all over the world. We are all working together, trying to cause change, trying to see if we can edit the genomes. And the future ahead is really bright. So what else can we tackle? Can we go to sickle cell anemia? This is a girl called Nivea looking for marrow donation. Can we move on to cancers? This is a guy that died last month in Glasgow from cancer, 29-year-old with a son. Can we move on to hemophilia? Can we treat these diseases by cutting out regions of their genome? Or can we just go to other genetic conditions I'm not mentioning, but that you know, or that someone around you knows, or that someone in your community knows? Or can we go to all the viruses and all the pathogens of the future? Last year, there was a big scare about Ebola, which is very similar to HIV. I'll just hand like almost every scientist does with something cheesy. So if we knew what we were doing, it would not be called research. Thank you.